Hey, Bob class. This is your section 4 notes, Introduction to Genetics. We're going to go through the process of meiosis. A little bit different than mitosis, but definitely has some similarities. So we'll talk about those in this section. Buckle up. So as always, I like to throw a few learning objectives up. Don't need to write this down, but as you progress through these notes, you should be able to differentiate between the word diploid as well as the word haploid based off of the number of chromosomes. From there, you're going to identify and understand each step of meiosis 1 and 2. So it's almost like we're going through mitosis twice. It's going to be kind of a big deal, but I'm going to go through it as quickly as I can. And you should also understand the production of spermatogenesis and oogenesis, which is how we make sperm, and how we make eggs, which is pretty much the main reason why our bodies go through meiosis. And last but not least, can you differentiate between mitosis as well as meiosis? Well, let's get after it. So first thing I want to talk about is something called chromosome number. We've discussed it a little bit in my class, I know, but to kind of help explain this process, we're going to talk about fruit flies, aka Drosophila. As you can see in the top right corner, that's a fruit fly. They have 8 chromosomes, which means 4 came from mama and 4 came from dada. And these two sets are considered homologous, meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. This would be just like our blood groups questions when we did our Punnett squares. If you have an AA mixed with a BB and you make AB, that means you got a homologous chromosome from mom and a homologous chromosome from dad, and you got A and B, which gives you AB blood type. So homologous just means the same, equal sets, and this is how we get our different alleles. This is how we are, genetically different from everybody else. Here's a couple things to look at. Through this process, the whole goal of meiosis is to have genetic variation, which means you're going to be a little bit of mom and you're going to be a little bit of dad. Some things that I want to point out to you is that if we look at a homologous chromosome here, we'll say this we got from mom, this one we got from dad, and it is homologous, it's the same. And we go through this process called crossing over. And basically this is what you need to see, is that they kind of swap some genetic information. There's this little portion here that was once red moves over to the blue one, this one that was blue moves over here. And it kind of recombines the chromosome sets. So this is one way through crossing over that we can get some genetic variation and get you to look a little bit like mom and a little bit like dad. So let's define some words when we're talking about chromosome numbers. A cell that contains both sets of homologous chromosomes is said to be diploid. And diploid means, di means 2, so you have 2 the sets. And it's sometimes presented as what we classify as 2n. So for instance, Drosophila fruit flies 2n equals 18. So that's saying a full set of 8. A full set of chromosomes is 8. For humans, a full set of chromosomes would be 46. Okay? And what that means is we have 46 total chromosomes. This is a human chromosome being sequenced. And you'll notice that we have 1, 2, 3, 4, 5, all the way down to your X and Y, your sex chromosomes, which is 23. But they are 23 pairs. 23 times 2, bing, 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 46. Dogs, their full set is 78. So what this means is you take any cell that's not sperm or egg in your body, skin cell, nerve cell, and we sequence the amount of DNA, it'll always be this. 46 total chromosomes in a skin cell, 46 total chromosomes in a nerve cell, okay? 23 pairs in each cell, okay? So in a full diploid cell, we have all the chromosome information, which means we have every blueprint to make anything in the human body in any one of our diploid cells. So by contrast, sexually reproducing organisms contain only a single set of chromosomes called haploid. I always think of haploid as half, haploid half, which means one set. So for fruit flies, diploid was 8, N which we classify as haploid, would be 4, half of it. For humans, N would be 23. For dogs, N would be 36, which means that our sperm and our egg actually contain half of the necessary genetic information. And when sperm and egg meet and it makes you, one cell, that's where we recombine and get to that 46 number. All right, put your seatbelts on. We're going to fly through meiosis now. First, I'm going to define it, and then I'm going to quickly go through each step. I recommend jotting, maybe a simple picture down as we move through this, but other than that, it's just going to be straight up info. Meiosis is a process of reduction division, which means we're getting smaller, in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. What does it mean? Here's a cell. We are going to replicate it like we used to do, and then we are going to break it down into 4 essentially in males haploid cells, and 2 as you can see are actually what occurs in mitosis. Okay, so 2 identical cells. Meiosis is divided into 2 distinct divisions: meiosis 1, meiosis 2. And as you can see here, meiosis 1, meiosis 2. The goal, at least for sperm and egg, is to make 4 haploid cells. So 4 cells with half of our genetic information. All right, so here's our first one. Prophase 1, and folks, it goes in the same order: PMAT, PMAT. In prophase 1, each chromosome pairs with its corresponding homologous chromosome to form what we classify as a tetrad. So our chromosomes pair up, just like they did in prophase 1. Notice the nuclear membrane is breaking down as well, but at this point, portions of each chromosome swap. And this is really, really important to know. This causes crossing over. I talked about that on the previous slide. And this is our first step into getting some genetic variation in you. So if there are specific alleles like red hair, blonde hair, brown hair, strawberry blonde, auburn, that is an allele. This is where we could possibly pass on that genetic variation. All right, metaphase 1. Spindle fibers attach to the chromosomes, and they line up individually and randomly on the metaphyseal plate, like you see here, just like in mitosis. This random alignment is called independent assortment, which means that they land, there's no necessary spot for you. It's not like a seating chart in this classroom, okay? You walk up to that metaphyseal plate, you might have started off on the right side and now you're on the left side. Doesn't really matter. They line up randomly. And if they line up randomly, when they go to get split, remember in anaphase where these chromatids get ripped apart, if they lined up randomly, that means some of that information is going to go to a different side. And that is going to be classified as independent assortment. And this allows for further genetic variation. So that is now 2 ways that we have genetic variation: crossing over and independent assortment. All right, next one on the list for meiosis is anaphase 1. Very simple. The fibers pull the homologous chromosomes toward the opposite ends of the cell. Please, please, please notice there's an example here of crossing over from prophase 1, and now that information is being pulled to opposite sides. After anaphase, we go into telophase. Nuclear membranes form up. The cell separates into 2 cells. As you can see, we'd then go through cytokinesis, and we'd have a little bit of split here, but notice our genetic variation. From there, we go into what we classify as mitosis 2. We go through this process twice. Prophase 2, meiosis 1 results in 2 haploid N daughter cells, each with half the number of chromosomes as the original. As you can see here, we had 4 in prophase 1 and prophase 2. Now we have 2 cells with 2, or half of the chromosomes that were before. Okay, so this was a diploid cell. These are 2 haploid cells. They have half the number. 4 chromosomes in here, 2 in this one, 2 in this one. Hopefully that makes sense. In metaphase 2, the chromosomes line up in a similar way to metaphase stage in mitosis. Line up on the middle, as you can see again. Only difference is we still have haploid cells. Okay, not as many chromosomes there to separate out. Anaphase 2 is the same. Spindle fibers attach. Sister chromatids move in opposite directions. As you can see here, here's our differences. Again, major drawback, or not major drawback, major takeaway. Half of the chromatids are being moved apart because we've split that into 2 cells. Ooh, I sound like a broken record here. Telophase 2, meiosis 2 results in 4 haploid end daughter cells, as you can see here. In telophase 1, we ended with 2 cells with half the information. Now we have duplicated each of those cells, kind of going through what we classify as a mitosis phase. And now we have 4 cells with half of the genetic information. So we started out with 2N, and now we have 4N. So out of one mother cell, we made 4 sperm. We made 4 eggs, all with half of that information. So in males, haploid gamete formation is called a sperm. And as you can see here, the process of spermatogenesis, genesis means the beginning. So the beginning of making sperm, we break apart into 2, then into 4. All of these have half of the information inside of them. Spermatogenesis will make 4 sperm from 1 cell. On the opposite end, for females, in females, haploid gamete formations are called ovums or eggs. Oogenesis is the process, and it does produce 4 cells, but it only makes 1 egg. And this is what's unique about oogenesis, is that yes, you have made 4 cells. 1 of those 4 is going to become the egg, and the other 3 are classified as what we call polar bodies. And all these are going to do is they're actually going to be used to feed the major egg. So unlike sperm, where we make 4, we do make 4 eggs, but 1 actually is going to mature into the ovum, and those other 3 eggs, quote unquote, are going to be used to feed it. So it kind of eats other eggs. It's kind of cannibalistic, but got to feed that egg. It's hungry. All right, you made it. This is our last slide. This is a busy unit. I want to take a moment to compare mitosis to meiosis, and maybe you just want to put this chart in your notes. Maybe you don't want to write that down. I think this chart would be perfect to put in your notes because what it describes is we take a diploid cell in mitosis and we make an identical copy, 2 diploids. In humans, we have 46 chromosomes. So in mitosis, you're going to make 2 cells, 46 chromosomes. Meiosis is different. Okay, we go diploid to haploid, and 1 diploid cell is going to make 4 haploid cells. We're going to split it in half, and then we're going to duplicate that half. Okay, a lot of people get confused right here. But bottom line, what I want you to know, mitosis we make 2 identical cells, meiosis we make 4 cells with half of the genetic information. As always, I throw some review questions in here to make you think a little bit about what you've done today. Wow, it sounded very parental, like I'm yelling at you, but yeah, think about what you've done today. Take a moment. I'm not going to answer these questions, but you should be able to based off your notes. If you have any questions at all, feel free to see your local biology teacher, myself included. Other than that, have a fantastic day, and we will catch you on the flip side. Yago out.